Living things are very diverse and are found in an incredible array of colors, shapes, and sizes. Yet, in spite of their great differences, if you look closely enough, they are actually quite similar to one another. As long ago as the 1830s, scientists had begun to realize this fact when they discovered that all living things are made from cells. By the middle of the 20th century, even more similarities were found when new scientific tools let researchers examine the molecules found inside of cells. One molecule in particular, called deoxyribonucleic acid, or DNA, captured their attention. Scientists discovered that DNA uses a genetic code to chemically store the information cells need to perform their many life-supporting activities. Researchers also discovered that this genetic code is truly universal because it is used in every living cell, from lowly bacteria like these to the complex cells of animals and plants. Now let us learn more about the amazing DNA molecule and find out how it works. In the cells of higher organisms, almost all the DNA is located inside the nucleus in the substance called chromatin. Before a cell reproduces, the chromatin changes into the separate structures seen here that are known as chromosomes. Inside a chromosome, the DNA, along with some proteins, is neatly packaged. It is twisted and folded over and over again. Something like this is possible because DNA is a very large molecule, a macromolecule. A closer look shows that the DNA molecule is actually made up of two separate strands that wind around and around one another, creating a shape called a double helix. Each of the DNA strands is made up of smaller chemical subunits called nucleotides, which are joined together one after another in a very precise order. Only 4 different subunits are used in DNA, but each one can be found tens of thousands of times in the huge molecule. To help visualize molecules, scientists use structural formulas like this one to show how their atoms are bonded together. For example, this structural formula shows that each DNA subunit contains a phosphate group composed of a single atom of the element phosphorus, P, bonded to 4 atoms of the element oxygen, O, plus a single hydrogen atom, H. Each DNA subunit also contains a sugar group called deoxyribose that is made from 5 carbon atoms, 1 oxygen atom, and several hydrogen atoms, all bonded together as shown here. The individual DNA subunits in a strand are linked together by strong chemical bonds between their phosphate and sugar groups. These bonds form the backbone of the DNA molecule. Each of the 4 DNA subunits possesses a nitrogen-containing base. The chemical names of the bases are thymine, cytosine, adenine, and guanine, abbreviated T, C, A, and G. These bases are always found in pairs linked to one another by weak chemical bonds called hydrogen bonds that exist between the bases in each strand. Bases always pair up the same way, so that if one DNA strand has an A, the opposite strand will always have a T in exactly the same place. Likewise, a G in one strand is always paired with a C in the same place in the opposite strand. Because of the way base pairing occurs, DNA molecules end up having 2 exactly opposite or complementary strands. As we will soon discover, it is the precise order the subunits appear in the DNA strands that is the key to the genetic code. And the genetic code is the key to life itself. In the last century, researchers studying genes, the units of heredity which determine such things as hair color, eye color, and blood type, made two extremely important discoveries. The first was that genes are made from DNA. The second was that each gene holds the coded instructions for making a different protein. Proteins are made up of long chains of amino acids, of which there are 20 different kinds. Living organisms use proteins for a lot of different things. They can be used for building cell parts, repairing damage, fighting disease, and regulating the speed of the tens of thousands of chemical reactions that take place inside of cells. Each chromosome in a cell has many different genes, and genes themselves are simply certain regions of a DNA molecule. Genes determine the order amino acids will appear in a particular protein chain, and it is this order which gives the protein the ability to do its job. Now let us see how DNA stores instructions in the unique biological language of the genetic code. The subunits of a DNA molecule, A, T, C, and G, are the 4 letters of the genetic code alphabet. Of course, the subunits are chemical compounds, not actual letters. Nevertheless, they can be used just like letters to spell out genetic code words. Most of the 64 code words used by living things represent or code for amino acids, and each of these words is only 3 letters long. For example, AAA is a code word for the amino acid phenylalanine, GCT for the amino acid arginine, CTC for glutamic acid, and TAC for the amino acid methionine. A protein made from these amino acids could have a gene or sentence of code words like this written in one of the DNA strands. This sentence informs the cell that this particular protein is to be made using only these 4 amino acids in this exact order. All the DNA in a cell can be thought of as a library containing books of instructions which tell the cell how to make the proteins it needs to stay alive. In fact, every time a cell divides, its DNA is copied or replicated, so that each new cell will have a full library of instructions. Let's briefly review how DNA stores instructions. The DNA molecule of a single chromosome can be thought of as one book from the cell's library of instructions. A chromosome can contain thousands of genetic code sentences or genes that determine the order amino acids will appear in proteins. Such sentences are written using genetic code words, all of which are just 3 letters long. These words are codes for any of 20 different amino acids, and of course, the letters with which the words are written are the 4 nucleotide subunits of DNA. Inside living cells, the actual process of making proteins involves several different steps, because the instructions for making proteins are stored in the nucleus, but the proteins themselves are made, synthesized, in the cytoplasm. This means cells must have a way to move information out of the nucleus and into the cytoplasm. As it turns out, cells use a special molecule for this task, called messenger RNA. RNA, or ribonucleic acid, is very similar to DNA, but RNA has only a single strand. It contains the sugar ribose instead of deoxyribose, and a base called uracil replaces the thymine found in DNA. So the RNA alphabet is A, U, C, G, not the ATCG used by DNA. Whenever a cell needs more of a certain protein, it sends a chemical signal to the nucleus, which causes the gene for that protein to be switched on. When this happens, the DNA code words in the gene are copied into the RNA code words, or codons, of messenger RNA. This process is called genetic transcription. After the gene is copied, the messenger RNA carries the protein-making instructions to a ribosome in the cytoplasm. Ribosomes are the organelles where proteins are made. However, just the fact that the messenger RNA has reached a ribosome is not enough for the protein to start being made. In order for the message to be translated, a third kind of RNA, called transfer RNA, is needed. Transfer RNAs are small RNA molecules that can pick up amino acids and transfer them to the messenger RNA on ribosome. There are almost as many transfer RNAs as there are DNA code words. In fact, each of the different transfer RNAs has a different anticodon, which is basically a DNA code word written using the letters of the RNA alphabet. Transfer RNAs carrying amino acids match up their anticodons to the messenger RNA codons as the ribosome moves along the message. As a result, the amino acids carried by the transfer RNAs will be put into the protein chain in exactly the right order. Once the protein is finished, it is ready to perform its job in the cell. And as the protein goes to work, a new chemical signal is sent back to the nucleus that switches off the gene for that particular protein. As we have just learned, DNA performs an essential role in the lives of all living things. It stores huge amounts of information in the unique biological language called the genetic code. DNA's genetic information is transcribed into messenger RNA, which carries it to the ribosomes where, with the help transfer RNA, new proteins are made to perform thousands of tasks inside of cells. But DNA is even more amazing because it can make exact copies of itself. As a result, genes and the instructions they hold are passed on, not just from old cells to new cells, but from one generation to the next. True or false? The 4 subunits of DNA are used as the letters of the genetic code alphabet. True or false? Genetic code words represent different proteins. True or false? Translation takes place on ribosomes. True or false? There is usually one gene on each chromosome. True or false? DNA is transcribed into messenger RNA in the nucleus.